Hi, everyone. We're just going to wait a minute to let everyone in, and then we'll go ahead and get started. All right, I think we can go ahead and get started now. Hi, everyone, and welcome to day 4 of Immunoassays Applications Week. Today, we have Dr. Rwik Sen here, who is from Active Motif. Rick will be talking about how to optimize your pull-downs for chromatin immunoprecipitation. He is a senior field application scientist, so he has quite a good amount of training on how to, train researchers, and he's also comes with quite a bit of knowledge about applications that are used in epigenetics research, such as ChIP. I also just want to make a quick note that this session is being recorded, and we will be sending a follow-up email next week to all registrants with a link to access the recording. But before I go ahead and pass it off to Rick for the workshop, I just want to share a few slides with you guys about Proteintech in case you are unfamiliar with who we are. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass over 13,000 targets. We also offer ELISA kits and IHC kits, as well as antibody labeling kits and cell separation kits. Within our Proteintech brand, we also have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins as compared to, for example, E. coli-derived proteins. We also have our Chromatech branded nanobody-based reagents. Nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity. And because of that, this makes them ideal for things like immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Proteintech known as Proteintech Genomics. And Proteintech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. As a company that is driven by scientists, we're here to support our researchers and scientific community any way we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. We also know the importance of product validation and offer open access validation data. When testing new products, we validate with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications and can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform that we use to manufacture our UniRab recombinant antibodies. With 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world so that you can get started on your experiments right away. As a company, we measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company's history, our products are being featured in more and more publications each year. And as of now, we have over 280,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. So with that, I'd like to go ahead and introduce our speaker for today, Dr. Rwik Sen. So Rick is a senior field application scientist and leads the equipment portfolio at Active Motif. He earned his Ph.D. at Southern Illinois University and did his postdoctoral training at the University of Colorado Anschutz, and also was awarded a fellowship from the American Heart Association. Rick's expertise includes epigenetics, CRISPR, single cell, and development. He has quite an impressive scientific footprint with over 30 peer-reviewed publications and book chapters, and also has a passion for mentoring STEM trainees. If you do have any questions for Rick during his presentation, please do feel free to put those into the chat or the Q&A box, and he will address them at the end during the Q&A session. So with that, I will go ahead and pass it off to you, Rick. Thank you so much for being here today.
Thank you very much, Afrida, for the very generous introduction, and thank you, Proteintech, for this opportunity to present. Today, I'll be going over ChIP, that's chromatin immunoprecipitation, and what we do as the leader in the field of epigenetics for the past 25 years. And also, I'll touch upon several different topics here, but the goal is for you to understand and appreciate the importance of ChIP and then be in touch with us as and when you are going to use this technique in your research. So we definitely look forward to, working with you and just helping you. That's our main goal here. So, and if there are questions that I can't answer right away today, I'll make sure to take them down for you and get back to you with answers. So with that, I'll start the presentation. And here, Afrida, can you see my slides?
Yes. It's not on the slideshow just yet, but it's
Yes, yeah.
see the slides. Yep, perfect.
Okay. Great, thank you. All right, so here's my email and LinkedIn, and if you have questions, you can also reach out to Afrida and she can pass them on to us. So this is the agenda for today, and I'd like to start with a very brief introduction of Active Motif. We've been around for the quarter of a century now, founded by Joe Fernandez, who was the co-founder of Invitrogen. And in 2021, we partnered with Proteintech. And in addition to all our products and services and equipment, we are very deeply engaged with academia. We support a lot of big and small academic events, and we have several resources, including grant competition on epigenetics, which we do from time to time based on the different services that we offer. We encourage you to listen to our podcasts, which are very interesting, and they are done by key opinion leaders across the world. And then we have so many peer-reviewed publications on people using our products and services. Now, coming to ChIP, this is our ChIP products portfolio, and you can see we have various different kinds of ChIP kits and also alternatives to ChIP, which are cut and tag and cut and run, which I will introduce at the very end of this presentation today. And then we have also another suite of chromatin analysis kits and then spiking. So as you can see, we have ChIP, cut and tag, cut and run. These three are the complete suite of applications that have similar readouts. And all three are very popular in understanding the interactions between chromatin and proteins. And then we have several other items, as you can see here. And if you go to our services webpage, you'll see that ChIP is on top, and then we also have all these different kinds of services. And the services are end-to-end, which means we do everything wet lab, the sequencing and the bioinformatics. And we also offer sequencing and bioinformatics for people using our kits. Then we would also do validation if needed and custom services as well. So because this is about ChIP, and I realize some of you may be new to ChIP, new to epigenetics, I will just give a very brief introduction of epigenetics here. So I like to talk about epigenetics to people who are new by giving the very relatable example of identical twins. And as you know, the identical twins have identical genetics, yet they are different in terms of the way they are susceptible to diseases, to obesity, infection, and everything. And epigenetics accounts for a lot of that. So epigenetics is the software, the different softwares that are run on identical computers with identical hardwares. Hence, the computers run differently. So gene regulation by epigenetics is done differently here, but the underlying genetic sequence is not changing. That remains the same. And epigenetic changes or regulations are also heritable. It's important to understand epigenetics because FDA has several approved drug candidates, therapies, which are epigenetic targets, and there are many, many clinical trials going on across the world on epigenetic targets. And beyond the biomedical scope, epigenetics is also everywhere in nature, from the giants to the tiny honeybees, which are so important for our ecosystem, as well as the food that we eat. So with that, I'd like to get into the complexity of the epigenome. If you look at one human, the number of organ cells and the DNA contained in the cells, chromosomes, RNA, proteins, everything that you see here, these are being regulated at some level by epigenetics. Hence, our health and disease, our mutations, our future generations, and our different stages of development, aging, everything is getting regulated by epigenetics. So if you look at a typical eukaryotic cell, you have your chromosomes in there, and they are made of chromatin, which is a mixture of DNA and proteins, very simply put. And the proteins are actually histone proteins. So the fundamental unit of chromatin is known as nucleosome. The nucleosome is 8 units of histone proteins, 2 copies each of histone H2A, H2B, H3, and H4. And 146 base pairs of DNA go around that histone octamer or unit of 8 histones. And that together form the nucleosome, which are the fundamental unit of chromatin. Now, it depends on whether the nucleosomes are closely stacked against each other or if they are spaced out with a lot of free DNA available for transcription factors to access them and cause gene expression to go up. So the nucleosome, this chromatin accessibility is a major factor for gene expression to occur. And that is one of the main focuses of Active Motif and one of the major epigenetic contributors. Now, how that happens is the N-terminal tails of the histone proteins made of amino acids, specific amino acid residues undergo specific modifications, like a methyl group or an acetyl group may come and bind to that amino acid residue, or it may go away. And there are several others, other such modifications. So all of these histone modifications cause these epigenetic changes. Then there are other enzymes which are ATP-dependent chromatin remodelers, which also cause epigenetic changes. And then there is DNA methylation, which is direct methylation on the DNA that is also contributing to the epigenetic changes. So these 3 are the major epigenetic factors or epigenetic contributors that Active Motif portfolio focuses on. Other than these, you also have non-coding RNA, which regulates RNA translation at the RNA level, or prions or protein modifications, which are also causing epigenetic changes. However, RNA and protein are not the main focus of Active Motif right now. So today I'll be talking about how you use ChIP or chromatin immunoprecipitation to understand the epigenetic regulation of gene expression at the chromatin level. So ChIP is not a standalone technique, and I wouldn't just talk about ChIP here because I want to give you the full context as to why ChIP is important and why you would use this in your assay. So when we help people design an epigenetics project, we always start from the basic assay that is on your left. You're looking at the chromatin accessibility. So you see, I was talking about the chromatin organization, whether the nucleosomes are close together, where you see this red region is inside, which is not accessible to the transcription factors, versus here, you see the relaxed chromatin, this red region is exposed. It's accessible to transcription factors. So depending on your experimental model, it can be disease versus mutation, it can be drug treatment versus control, or wild type versus mutant, or it can be different stages of development. So whatever your situation is, you would want to know whether there is a change in this chromatin landscape, whether the chromatin is open or closed. That's your first very basic experiment. So for that, you would do ATAC-seq. And ATAC-seq is the assay which will give you genome-wide regions of open chromatin maps. So we have kit and service both for that. And I also go out in the field and teach people how to do ATAC-seq. So once you are you get a clear idea that yes, my model is actually showing a change in this chromatin landscape, then you would want to go to the next mechanism, the targeted methods. What's the mechanism? What is happening in those regions of open chromatin? So from ATAC-seq, there is a bioinformatics process called motif analysis. The software will give you a computational list of the transcription factors which are binding to the motifs that are accessible or open. So from there, you can pick your transcription factors of interest and go to the mechanistic methods. And then you would want to do the method ChIP. ChIP will tell you genome-wide where your transcription factor is binding. And hence, this is how you tie in ATAC-seq with ChIP-seq, the discovery assay with the mechanistic assay. Also, you can look at the motif, the methylation or acetylation or various histone modifications. So in ChIP, it's not only transcription factors, you can also detect where your histone modifications of interest are binding. So Active Motif has ChIP, cut and tag, cut and run to help you with that part of the project, both kit and service. And then you can also do DNA methylation. You can add RIME, which is mass spectrometry-based chromatin immunoprecipitation to understand protein interactome, Hi-C for three-dimensional chromosome interactions, and finally complement all of that with RNA-seq. So this is the entire suite of assays that you would ideally want to do to get the full complete picture of what's happening epigenetically here. So any one assay here is not enough. They are all parts of the puzzle. So now today, I'll talk about like the ChIP, but then what are the applications? So mostly it's different aspects of biomedical research, but also a lot of emphasis on plant and animal health here. And then we have already expanded into single cell. We offer that on the 10x Genomics platform, but we also have these other interests in mind right now. And with that, I'd like to start with ChIP. So ChIP is done to investigate the protein-DNA interactions across the whole genome. And as you know, our genome, the DNA that's contained in the cell is 2 meters long. And there are several proteins that are interacting with that DNA. So the idea is to map those regions of the genome where your protein of interest is binding. However, when you start doing an experiment like ChIP and subject that sample to a lot of harsh conditions, the very first thing you want to make sure is that the proteins that are bound to the DNA don't fall off. They are bound there intactly. So to do that, we will first start with cross-linking our sample. So you collect your sample and then you cross-link. If you're interested in the chemistry, here's how it's done, but just mainly I want you to focus on here's your protein and here's your DNA. So you want to see, you want to make the interaction strong. So you add the chemical formaldehyde, and this is your cross-linked product that happens here. Now, once you're cross-linked, you quench by glycine, and then you either freeze your sample or you directly move on to the experiment. So then the first thing is to lyse, because you have to lyse and get the nuclear extract out, because everything you're interested in is inside the nucleus. And then, as I said, the DNA is 2 meters long. You want to cut that DNA down to workable size fragments, and that is done by sonication. So after sonication, you take 2 aliquots. So the first aliquot would be an input control, we call it, where you do not add any antibody. And then the other one is your IP or immunoprecipitate, where you would add your antibody of interest. And the purpose of having an input is at the very end, when you analyze your results, you would compare the signal that you get from IP versus the input. And that is why you need an input control. So once you got your sonicated fragmented chromatin, you can just schematically imagine that they look like this. There are several pieces of DNA with the optimum size is 300 base pairs, and the DNA and the proteins are all bound to different proteins bound to different pieces of DNA. Now, if you are interested in this green protein here, for example, let's say it's MYC. So you add your MYC antibody. This is the recommended amount of antibody to chromatin that you have to add. And then if the antibody is very specific, so that's very crucial to have a specific antibody with no background and no off-target effects. So that should then ideally just bind to the MYC proteins on your pieces of chromatin. No off-target or non-specific bindings. And then you would add beads to immobilize the immune complex, and everything else is washed away, it's gone. And then what you do is this. So you purify that piece of DNA that was immobilized and do NGS library prep, sequencing, and bioinformatics. Now, when you finally get the reads, this is an example of how you interpret that. So those reads are bioinformatically aligned or mapped to the reference genome. So if the reference genome here is the gene ACAP2, you find that your reads that you got are aligning or mapping to this part of ACAP2. That means your genomic loci 1 and 3 that you got from here are these regions of the gene. So what this means is this green protein, MYC, is binding to these two regions of your gene ACAP2. So this is the overview of the workflow of ChIP, and this is how you interpret and analyze the results. Now let's get into some more details of this. So as I said, the antibody is very crucial for success of ChIP. The other part, which is very crucial, is the fragment size. If your fragment sizes are not of optimum length, they will not be properly immunoprecipitated or pulled down. Hence, you need to have a very consistent method of sonication, and for that we have the Pixel platform. If you're interested, we are happy to talk more about this. And then we use very precise equipment like Agilent fragment analyzer or tape station. We have tech notes on these on how to, process the sample through Pixel and then measure them through here and then get very clear, precise results. And it's not only for ChIP, Pixel is also integrated with all these other kinds of workflows, including proteomics and all these different kinds of samples can be processed here. So now coming to the cell fixation or cross-linking step. So this is available on our website. Everything that I'm showing here today is publicly available, so we are happy to share all these resources. So this is how we advise people to prepare their samples and then ship them to us for doing ChIP-seq. So mainly you will need formaldehyde, and then everything else that you can see here, these are all the different buffer components. And you will fix this with exactly this, a very precise percentage of formaldehyde, no overfixation. And the time is very, very critical. If you are handling multiple samples, have a lab member help you to maintain the exact time for each sample here. So first do the formaldehyde and then do the glycine and then finally the PBS-IgE-PAL. This helps to not have the cells stick to your tubes, and hence you don't lose samples, because if you're working with rare samples, that's very critical as well. And then finally you do this PNSF or protease inhibitor treatment here and then freeze them at -80, or you continue with the experiment. So this is how your fixation is done. And this is, again, a publicly available document. If you are working with tissue sample prep, then we just recommend sending the frozen tissue to us, either in dry ice, liquid nitrogen frozen, and we do the rest. But if you want to do the tissue processing at your end, then you may want to use this Omni beadruptor or some instrument like this, because that helps to homogenize the tissue before you start sonicating the tissue here. And here are some tips that we recommend people do not overfill the tube and seal if you think it'll be transported or anything. And of course, labeling and documentation are very important for the tissue type here. So the starting material, it can be cell tissue, fresh, snap frozen, FFP. Regardless of whatever you're using, quality is very important. So quantity and quality. The quantity, we recommend 4 million or above for transcription factors. For histone modifications or RNA polymerase II, which are very robust, you may get away with 1 million or so. Now, it is true that people have been optimizing ChIP for 100,000 or in that range, 100,000, 500,000 range. So if there are concerns and if you don't have enough samples, and that's one of our biggest concerns that we get, feel free to reach out to us and we have some recommendations on those. We have some alternative methods that are soon to launch, which can process as low as 100,000 cells for each sample. So we'll be happy to work with you on that. And then we aim for 30 microgram chromatin, and as I said, the 4 microgram of antibody is what we typically use, but then again, depending on the sample type and everything, we may need to change some of our protocols here. Now, sometimes you may want to look for cofactor interactions. So it's the cross-linking that we do is not always protein DNA. It can also be protein-protein because you have multiple proteins interacting on the chromatin. So there could be there could be transient interactions, not very stable or very low expressing transcription factors or indirect DNA binding. For that, you can do a double cross-linking. So first you cross-link with DSG, and then for 30 minutes or 45 minutes, and you get a lot of protocols on that as well. And then you do the formaldehyde cross-linking, and then you go on to the rest of the experiment. So these are some other methods that you can implement for that. So now coming to immunoprecipitation. So you did your cross-linking, quenching, lysis, sonication. Now you will do the immunoprecipitation, which is adding your antibody of interest. So again, very specific antibody. You look up all the different vendors whose antibody has been tested for ChIP, validated, you see good validation results, then proceed. If you don't find one that is validated, we run validations for the customer as well. And then you can also, the other way can be epitope tagging your target. I used to do ChIP on yeast cells, and a lot of times epitope tagging helped, and it was easy to do epitope tagging on yeast as well. And then during my postdoc, it was zebrafish, and then that was a little challenging to find antibodies for zebrafish. And then you would validate your sample. So there are methods which you can use to validate here the antibody, and then finally the sequencing. So before you come to the sequencing, the steps that I showed in that schematic, like you have to do all the DNA purification and then the PCR and the library prep and finally the sequence. So these are what we recommend, but you can see in the literature there will be variations on this, but typically it's something around this. But the main thing here is it requires quite deep sequencing, so 30 million. The alternative methods, cut and tag, cut and run, they are much shallower sequencing. Okay. So I'm not a bioinformatician, but I have put some slides together for you just to give you an overview of this, because at this day and age, we all need to have some understanding of bioinformatics analysis, because sometimes the bioinformaticians don't have a very good understanding of the biology side. So we need to work together to make sense of the data sometimes. So these are some of the steps that we do, and I'll go through these. So first is adapter trimming, because when you did the library prep, you added adapters and everything. So you need to get rid of those, and then finally look at the reads, the sequences that you got here. And all these softwares are what we use. These are open source, so everybody can access them and use them. So we use the Illumina sequencing, and this is a very nice video on YouTube that you can find on the Illumina channel. It's called Overview of Illumina Sequencing by Synthesis Workflow. And you can also look at this publication here. So basically this tells you how the sequencing is done. So you have your fragments, get your adapters done, then you have an Illumina flow cell, which is a glass slide, where on this slide, you have these little oligos, and when you add your fragments, these go and complementarily bind everywhere on the flow cell. And then it's a process called bridge amplification, where the fragments would then bend like this and form like a bridge, and then the RNA polymerase II will come and the polymerase will come and cause the fragment to amplify here like this. So, and then they get separated, dissociated, and then they form again their own bridges. And hence, like this, you form different clusters, and then you get your signal by here. So the molecules here they will come and they will give you this like a light signal. So each base will have its own individual light, and from that you can scan the signal and find which base is here. So this is just a very basic overview of how this is done. You'll get the details and everything of how Illumina sequencing occurs. But this is basically for you to understand. So once you got your sequencer, the first raw data file that's called FASTQ file is what you get, and then you can process your FASTQ files to get all the different interpretations. So here's the flowchart. So you get your reads, and then you process them through different steps. And finally, you want to align them to your genome. Where in the genome did this read come from? And then you call the peaks. So you can, when the reads stack up and they meet certain criteria, that's a true peak. And then that peak means that here is where my transcription factor or histone modification of interest is binding. All of this can be accessed here in GitHub and other places. So, and these are again mostly all open source here. So this is how a FASTQ file looks like. This region here, the second row, is your actual read, and you have other components added to this. So this is like line 1 is a sequence identifier, and then this is your quality score. So as you can see on this little box here, you have a scale from 0 to 40, and it tells you where your different, like what your different characters mean, like what's the quality score. So if you have something from like here, from 20 to 30 to 40, any of these, that's a high quality score. So that's good. That means the accuracy of that is 99.9%. So here, this read is a good score because everything that you see here is on the higher end. If you had some characters here at this range, that would have meant it's a bad score. So this is how you analyze and interpret your FASTQ files. And then you do the alignment. So you already know your reference genome, can be human, can be any species. And then all your reads are getting aligned. And then, you see now that here is where my reads are aligning. So here you're dealing with two kinds of files called SAM file and BAM file. So SAM contains individual read information and genome alignment. And BAM is its binary version, which is not readable by humans. It's machine readable. So these are the terms you'll come across. Then peak calling. So once you do the alignment, you have some software to call the peaks as the true peaks versus not true peaks. So that's very critical. You have to set the parameters accordingly. Now, for example, if your peaks form here, that on this part of the genome, that means here is where your transcription factor of interest is binding. So you see your peaks are forming here and there. And then you would use the Integrative Genomic Viewer or IGV to actually visualize your results, the final results. So in the IGV, you can upload your files, and then it'll give you something that looks like this. And this is something, even if you don't do bioinformatics, you are familiar with, because this is what you see in the publication results. So at the bottom, you'll see the gene names. As you can see, it's TM, EM, and MET, and everything else. And you can see the different exons and other parts of the genome. Now, what these peaks mean here, this is a very interesting experiment where you're seeing a treatment by DMSO and a treatment by GSK inhibitor, and you see the ChIP signal for H3K27 trimethyl. And you can see that there is not much difference in this. So my idea for showing this slide is not only to explain how IGV works, but also introduce a concept which has been developed by Active Motif, Genentech, and Constellation, 3 different companies together. So first, let me give you a background of this experiment. So this is ChIP for H3K27 trimethylation. For those of you who are new, this histone modification is a mark of repressed transcription, so gene repression. And this is caused by an enzyme, EZH2. And if you inhibit EZH2 by GSK126, a chemical inhibitor, then EZH2 is inhibited, hence the repression is inhibited. That means, so you're repressing the repression, and that means it's activation. So H3K27 trimethyl should go down. However, you are not seeing that in this situation here. So why? Did you do your ChIP wrong? No. The answer is you missed something. You didn't do the experiment wrong. So what are you missing here? You're missing spike-in. So spike-in is the new technology that I mentioned just now. So here is your experimental situation. You have your human chromatin and target human antibody. You need to add spike-in, which is Drosophila chromatin and Drosophila antibody for histone H2AV variant in a specific ratio, which we recommend. Then you do the ChIP exactly in one single tube. So you're treating this as one sample. And we have done several validations and customers have done, they have published, there is no cross-reactivity between antibodies here. So they undergo ChIP. You take them through the full process. During sequencing, you map the respective species to the respective reads, and then you apply a normalization factor. And then after that, this is what you see, the true results. So you did the inhibition. Indeed, the inhibition is reducing the signal for H3K27 trimethyl. So this is your regular ChIP. This is spike-in. And this is the publication at the bottom where you can read more about this. So overall, when do you need spike-in? Is when you have a global inhibition, a global change, which you expect, but you're not seeing that in your regular ChIP. It's not for local changes, it's for global changes. And bioinformatics analysis alone cannot help you detect this difference. So you have to do the spike-in here. So this is the publication in PLOS One in 2016 between the three companies, and Brian Egan at Active Motif, he's the first author. Because we have time constraint today, I'll just tell you that there is a webinar on our website which you can access under the resources page and learn more about the spike-in in great details for not only ChIP, but also for these all other assays. Now, the other thing that we want to do with ChIP is motif analysis, which is very interesting because you want to know what transcription factor motifs are in those peaks that you detected. And this was developed by Dr. Benner at UCSD, and that's a few weeks back with him. And this is the link which you can access for the Homer motif analysis. Again, you can just Google Chris Benner, UCSD, and you'll find all of this information. And finally, the alternatives to ChIP, the cut and tag and cut and run. Here, the overall takeaway from these two assays is much less expensive, much less sample required. And you don't need cross-linking in any of them. So this is native state. You just need a different enzyme. So both are enzymatic. One of them is a TN5 transposase based. The other one is micrococcal endonuclease based. And then in the first one, because it's TN5 transposase, you just get your library. And then the other one, you have to separately make the library and do. Now, cut and tag is more favorable for histone modifications or robust factors because there are high salt wash conditions, which are not good for transcription factor binding. Cut and run is more suited for transcription factor detection. And you can do mild cross-linking as well for this. And I'm happy to answer more questions on these methods. And much less sequencing required, so much less expensive and low starting content and better resolution. The only thing is they are quite new, so not a lot of validated antibodies. The list is quite limited and no long-term data. And these were developed by Dr. Hennikoff at Fred Hutchinson. And again, you'll find a lot of interesting papers from his lab on these methods. And this is Dr. Hennikoff, where I want to highlight that he maintains a lot of protocols and does active question answer troubleshooting at protocols.io. So if you have questions, either we or great to reach out to him. So finally, things to remember, every step is crucial, but again, the antibody and sonication are more crucial than everything else. And then sample preparation will depend on the sample type, whether you need to do more double cross-linking or single cross-linking, how much lysis and all of that. And then also we are there with our entire ChIP portfolio to help you, a lot of educational resources and our tech support team. And with that, again, here's my email, here's my LinkedIn. We also have tech support, and stay tuned for all our promos and grant contests and everything. So with that, I'd like to thank you all for your attention and pass it over to Afrida. Thank you.
Great. Thanks so much, Rick. That was really a really useful webinar, especially for me,
Thank you.
coming from like an antibody company. I kind of stop
Mhm.
at the antibody part. So seeing everything kind of downstream was very, very helpful, and you laid that out really nicely. So thank you so much. We will do a Q&A session, but before we do that, I just have a few last slides that I wanted to share with everyone. So as Rick had mentioned, your antibody choice is going to be really important for your ChIP experiments. So I just wanted to share a few product slides that might be interesting for our audience today. So at Proteintech, we offer a very expansive portfolio of stringently validated polyclonal, monoclonal, and recombinant monoclonal antibodies that cover over 2/3 of the human proteome. We have over 300 antibodies that are validated in ChIP and over 4,500 that are validated in IP. Also, if your downstream application is Western blot, we do offer a very large portfolio of over 20,000 antibodies that are Western blot validated as well. Most of our antibodies are validated in multiple applications, so you can use one antibody for all your experiments. So for example, here on the right, I'm showing an example of our histone H3 monoclonal antibody, which is validated in IHC, IF, Western blot, IP, and flow cytometry. We have antibodies that also span many different research areas, including those studying, histones or histone modifications, DNA methylation, or RNA modifications. In addition to our traditional antibodies, Proteintech also offers nanotraps for IP and ChIP. Our nanotraps are nanobodies or VHHs that are extracted from heavy chain antibodies from alpacas. These nanobodies are then conjugated to different resins such as agarose, magnetic agarose, or magnetic particles. And then these nanotraps can then be used for IP and ChIP. So what makes our traps kind of ideal for pull downs is that because of their small size and because the beads are actually covalently bound to the nanobodies, once you actually elute your sample, the nanobody remains bound to the beads. So what this means is that if you run your samples after using a trap, so for example, if you're doing a Western blot and you run it on a gel, you don't have to worry about any sort of contamination of your heavy or light chains from your antibodies, which you have to worry about when you're using traditional antibodies. Our traps can also withstand really harsh washing conditions, even when you're using buffers with chaotropic reagents or high salt concentrations or with lots of detergents. The nanotraps are also, they have very high affinity for their targets, which leads to very efficient pull down, even if your protein is of low abundance. So you can see an example of this with our DNMT1 trap on the right here. So here what our team did was they performed an IP for endogenous DNMT1 from HEC293 cells, followed by Western blot for DNMT1. And so in the blast, what we're showing you is the first lane is basically your input, then the flow through, so basically what's left over after you've performed your IP, and then your bound fraction. And you can see that after using the DNMT1 trap, there is an enrichment of the protein of interest in that bound lane. So if you are interested in trying out our nanotraps, we do offer free samples of our nanotraps. So if you'd like to try one, you can request one on our website at ptglab.com under the nanotraps tab. Proteintech also offers antibody kits for those that are studying RNA methylation. So our RNA methylation antibody kits are curated packs that contain multiple trial size antibodies against key targets in that area. So this is going to include antibodies for writers, readers, and erasers, as well as antibodies against the modifications themselves. So for example, like M6A, M5C, M7G, and M1A. Because these kits are carefully curated and stringently validated, it saves you time from doing your own antibody searches. It also allows you to analyze multiple targets at one time, and it's also very cost effective because these are going to be your 20 microliter trial size antibodies. So it's going to be ideal for if you're, for example, starting a new project or if you just require a small volume. In addition to products for IP and ChIP, we also have several resources available on our website. This includes our blogs, such as 7 tech tips for successful ChIP experiments, or choosing the right antibody format for your immunoprecipitation, as well as many other blogs that we have available. We also offer protocol support and troubleshooting help. We have an IP guide, which includes a detailed ChIP protocol, and we also provide protocol videos as well. And then I also just want to highlight real quick a few more resources that we have available on our website. So you can check out our past webinars on our video hub. Our webinars cover a wide range of topics, including different applications and cutting edge research areas, as well as career development workshops that are hosted by internal and external speakers. We also recently launched our online learning portal. This portal offers free online training courses. So currently we have two courses on there. We have an image analysis using ImageJ and statistics for life scientists. Life scientists, sorry. Statistics for life scientists. It's a lot of S's in one title. These courses are really great because they're completely free. And once you've passed your course, you do receive a certificate. Also, today's workshop was a part of Immunoassay Applications Week. So every day this week, we have been hosting a workshop on a different immunoassay. And tomorrow, we are going to have our last day, and the topic will be immunofluorescence. So if you are interested in that topic as well and haven't done so already, please do feel free to sign up for our workshop tomorrow too. And then just lastly, I just wanted to make a note that everyone who registered for this workshop today will be receiving a follow-up email that will have a recording of this webinar as well as a promo code for 25% off one full-size primary antibody. You will also receive another email that will give you a certificate of attendance for this workshop. We do ask that you please allow 1 week for this email just because we're doing Immunoassays Week throughout this week, so we'll be sending out these emails early next week. So with that, I just wanted to thank everyone for joining. We'll begin the Q&A session now. If you do have any questions after the Q&A session is over, please do feel free to email us at events@ptglab.com, or you can email Rick directly at our sen@activemotif.com. So with that, we can go ahead and begin the Q&A. There were some great questions that were coming in while you were giving your talk. So the first one that I have up here is, how can you detect the interaction between long non-coding RNA and chromatin?
Yeah, I was looking up that question actually. So we first of all, I'd like to ask the audience if anyone has any inputs on that matter. The other one is we typically want to look at the proteins associated with the long non-coding RNA and whether they can be detected on the chromatin.
Okay,
But I'll make sure to. I've already actually reached out to our team for some of these answers here.
perfect, awesome.
Mhm.
Yeah, some of these might need follow-ups.
Sure, yeah.
The second question is, how do you overcome the low concentrations of ChIP-seq library samples? Do you have any general suggestions?
Yeah, it depends on the kind of the QC that you do on the libraries, what they look like. Because even if they are low sometimes, if the traces look good, then you might want to go ahead with sequencing. And it's not just for ChIP-seq, it's also for other assays like ATAC-seq and all. I'll almost always tell the people to send us, if they have any questions, to send us the results, and we can take a look, because not always do the traces look ideal and the concentration can be low. So we have to make a decision on a case-by-case basis for that. So just having low concentration doesn't always necessarily mean that it'll fail sequencing.
Okay, great, thank you.
Mm.
Our next question here is, someone says that I was looking for a kit or a way to quantify the activity of a certain histone methyltransferase. Is there a specific way to do it? What we did is Western blot of its corresponding histone modification, but we were looking for a more specific way.
So that's a great question. Afrida, is that more in your portfolio to understand protein activity assays? Like this
Yeah, so
Proteintech? Yeah.
I was going to say at Proteintech, we do have antibodies specifically for certain histone modifications. So, from
Mm-hmm.
kind of the antibody perspective, we would recommend doing Western blot as your downstream
Mm-hmm.
application. I just didn't know, Rick, if you had any other thoughts in terms of if there's another, more high-tech way of doing it, because I know Western blot is a little crude sometimes.
Right, yeah. I mean, we could always, so if we know what histone, it's a histone metal transferase. So if you know more about what that methylation does, so we could always do a like a knockout situation and maybe look at attack-seek or something like that, whether it's impacting the chromatin or it. I mean, I don't know how much is already known about that histone methylation. So do we already know that whether it's making a change in the chromatin landscape, or if that's already known, then do we know which genes it's targeting, and then we can move one step forward and do a ChIP on that to see whether any binding of any cofactor or anything is affected. Or we can also do an RNA-seq on that to see whether the gene expression levels are changing. So depending on how much is already known about it, we can look at all those effects of that or the roles of that metal transferase on the chromatin. I'm just talking from a chromatin epigenetics perspective. But again, there are more sophisticated assays just to look at protein activity or protein function. That's not directly in the portfolio of Active Motif.
Great. Thank you so much, Rick.
Thank you.
So there was one question. It seemed that someone was interested in doing ChIP or ChIP-seq with fresh frozen or FFPE tissues, and you had mentioned that you are able to do that. If they were curious about the cost, how would they find that out from you?
Sure. The kits are usually for 16 samples, and they cost somewhere between 12 to $1,600. And for the services, it's close to, depending on whether it's histone or it's a transcription factor, that varies by a few hundreds, but it's between 2,500 to 3,000 per sample. That's end to end, including all wet lab sequencing and bioinformatics per sample. And then depending on your experiment, we will also recommend one or two inputs. So we don't, and the inputs cost $1,750, and we don't recommend input for every sample here. What we do is, if you have 3 controls and 3 drug treatments, we would pool one input from all 3 controls and one input from all 3 drug treatments and then use them. So 1750 multiplied by the number of inputs, but the IP will be individually, so between 2,500 to 3,000.
Great, thanks.
Yeah.
The next question is, so they're considering using ChIP-seq versus cut and run with the transcription factor, which does not have an antibody that's validated. So for ChIP-seq or cut and run, can they try an antibody that's validated for IP?
Absolutely. Yeah. I mean, we do antibody validation. The first thing to remember is, regardless of whether an antibody is validated for ChIP, it doesn't mean it'll automatically work for cut and run. So you need to do validation for every new antibody that you're doing for cut and run. The second thing is, a lot of our customers and us, we have modified the original cut and run protocol to include cross-linking in the method and then get better success with a lot of these antibodies. But this cross-linking is much less stringent, or it's much milder compared to ChIP cross-linking. So a very low dose of formaldehyde for a very low period of time has helped with being successful for transcription factor cut and run. And again, happy to discuss that, the protocol with you in detail if you want to reach out. Yeah.
Yeah, and then also just a note from kind of the antibody side.
Mhm.
At Proteintech, if we do have an antibody that's validated for IP, you might want to reach out to us just to see if maybe we've had a customer who has run it in ChIP-seq or cut and run, because we might not have that internal validation, but it's possible that people have used it in the past. However, if we do have customer validation data, we generally put that on our website as well. So that's also a really good way of just looking at whether or not a antibody that's actually validated in IP might work for your particular experiment. And this is also a good opportunity for kind of experimenting with some of those trial size antibodies, so you don't have to make that investment on an antibody that might not necessarily work, but trying out maybe like a 20 microliter size antibody. So our next question here is, how can you break DNA to 1,000 BP by using sonication?
Yeah, with Pixel, it's very simple. You just we have programs for various specific sizes, so we can help you with that. Yeah.
Great. And then I think we have time for maybe a couple more questions. This question says, for ChIP-seq and cut and run, when we use E. coli spike in DNA, do we still need to use input DNA?
Well, E. coli spike-in is, I mean, Drosophila has worked better. When we were developing the spike-in technology, we have seen that, and that's one of the reasons being that Drosophila also has actual chromatin. E. coli doesn't have like true chromatin structure in there, so yeah. And we would recommend, if you are trying spike-in, we would recommend Drosophila just because it has worked best among the other things that we tried.
Great, thank you.
Mhm.
And then kind of the last question that I think might be nice to address is you kind of went over the workflow of ChIP. What type of controls do you recommend people use when they're doing ChIP experiments?
We used to do IgG control for a long time, but then we discontinued because it almost always gave nothing. So it was just an additional cost for doing that whole IP thing. So for a long time, we have been just doing input control for the chip. So there's really, I wouldn't say there is like a negative control. If you are, unless you are validating an antibody, that's a different thing. But generally, it's just your control batch versus your treatment batch, or wild type versus mutant or disease healthy, whichever situation that is. So that's been good enough. And ChIP is a very robust assay. It's considered the gold standard, has been here for decades. So that's all good.
Great.
Mhm.
Awesome. Thank you so much, Rick. And thank you everyone for joining. If we did not get to your question live, I'm very sorry, but feel free to email Rick directly at rsn, or you can at activemotif.com, or you can email us at events@ptglab.com and we can forward it over to him. Um, as I mentioned, we will be sending a follow-up email with a recording of this event if you want to rewatch it. So don't worry if you missed anything. And if you liked today's event and want to learn more about our upcoming events, please do follow us on social media because we post about all our upcoming webinars on there. So with that, I'll go ahead and end the webinar, and thank you everyone again for attending, and thank you, Rick, for your time and for the valuable insight.
Thank you, Afrida. Hey, Afrida, are these questions available like after the meeting? I see a
Yep,
Q&A box and a chat box, so both sets of questions.
yep, I can send them
Oh,
over to you.
great. Thank you so much.
Yeah, take care, everyone.
Take care. Bye.